Hello there, and welcome back to Sleepy Science Society. I'm so glad you found your way here tonight. Maybe you've had a long day, or perhaps you're simply the kind of person who prefers drifting off to the gentle rhythm of nature's deepest mysteries, rather than tossing and turning in silence. Either way, you're in exactly the right place. Tonight's journey is unlike anything we've explored before. We're diving into waters, both literal and metaphorical, to meet a creature so extraordinary that it seems to have discovered something humans have dreamed about for millennia, a way to cheat death itself. Before we begin, if these calm explorations into the wonders of science help you unwind at the end of your day, consider liking this video and subscribing to the channel. It helps others discover this quiet corner of curiosity, one sleepy soul at a time. And now, let's settle in and meet the only animal on Earth that might genuinely deserve the title of immortal. Imagine for a moment that you could press a reset button on your own life. Not metaphorically, not through meditation or fresh starts, but literally. Imagine that when your body began to fail, when illness crept in, when injury threatened, or when age finally caught up with you, you could simply rewind. Transform your entire being back to childhood. Start over with all the same genes, and live your life again from the beginning. It sounds like science fiction, like the fever dream of someone who's watched too many time travel movies. And yet, in the ocean's quiet depths, there exists a creature no bigger than a pinky fingernail that does exactly this, over and over, potentially forever. This is the story of Turritopsis dohrnii, the immortal jellyfish. And tonight, as you rest, we're going to explore every fascinating detail of how this tiny, translucent wanderer of the seas discovered a loophole in one of nature's most fundamental rules. The rule that says everything born must eventually die. The year was somewhere in the mid-1980s, and two young marine biology students had no idea they were about to stumble onto one of the most profound discoveries in modern biology. Christian Sommer and Giorgio Bavestrello were working in the warm waters around Italy, doing what marine biology students do, collecting specimens, cataloging them, observing them. Nothing particularly extraordinary about their day. They gathered some small jellyfish polyps, those early life stage forms that look more like tiny plants than animals, growing attached to rocks and surfaces underwater. They brought their collection back to the lab and placed them in jars, standard procedure. They watched as the polyps did what polyps are supposed to do. Over time, these anchored forms released tiny medusae, the free-swimming jellyfish forms that drift through the ocean like living parachutes. This was expected. This was normal. Everything was going exactly as the textbooks described. But then something happened that textbooks, at the time, said was impossible. The students forgot about one of their jars for a while. Not neglect exactly, just the natural chaos of a busy laboratory where dozens of experiments compete for attention. When they eventually returned to check on that particular jar, expecting to find either dead jellyfish or mature ones ready to spawn, they found something that made absolutely no sense. The jar was full of newly settled polyps, young ones, fresh starts. But there had been no spawning, no eggs, no fertilization, no larvae swimming around looking for a place to settle. The mature jellyfish had somehow transformed directly back into polyps. They had reversed their own development, moving backward through their life cycle like a film playing in rewind. For any other animal on Earth, this would be like a butterfly transforming back into a caterpillar, or a frog reverting to a tadpole. It simply doesn't happen. The arrow of life points in one direction. Forward, always forward. From birth to growth, to maturity to death. That's the rule. That's how biology works. Except apparently, it isn't. And sometimes it doesn't. The students continued to observe, now with the intensity of people who suspect they've found something extraordinary. They watched the jellyfish carefully. They stressed them in controlled ways, through temperature changes, salinity shifts, light starvation. And each time, when conditions became unfavorable, these tiny creatures would do the impossible. The medusa would sink to the bottom of the container. They would form into a blob of tissue, a cyst-like stage that seemed to dissolve all their structure. And then, within just 2 or 3 days, that blob would reorganize itself into a young polyp. A complete restart. A biological reset button. This discovery didn't immediately set the scientific world on fire. News traveled slower then. The jellyfish were obscure. The findings seemed almost too strange to be true. But eventually, word spread. Other scientists became intrigued. In the mid-1990s, at a marine biology workshop in Blanes, Spain, the topic came up in discussion. A researcher named Volker Schmid, who had spent years studying the cell biology of hydrozoans, which are the group that includes Turritopsis, was skeptical. He insisted it was impossible. Once a medusa reached maturity, he argued, the cellular differentiation was complete. There was no going back. To settle the debate, workshop participants, including a young scientist named Stefano Piraino, dove into the ocean, collected fresh Turritopsis specimens, and repeated the experiment right there. They stressed the jellyfish, waited, and watched. And just as the original students had described, the medusae sank, transformed, and produced polyps, right there in front of multiple witnesses, including the skeptic himself. The impossible was happening, repeatably, verifiably, undeniably. This wasn't a fluke. This wasn't misidentification. This was a genuine biological phenomenon that broke all the rules. And thus began the real scientific investigation into what would come to be known popularly as the immortal jellyfish, though scientists always prefer the more measured term. Biologically immortal, a crucial distinction we'll explore later. So, what exactly is this creature that we're talking about? Let's meet Turritopsis dohrnii properly, in all its tiny, translucent glory. If you were to encounter one of these jellyfish in the ocean, you might not even notice it. Not because it's hiding, but because it's so remarkably small. A fully grown adult Turritopsis dohrnii measures about 4.5 millimeters across. That's roughly the diameter of a pencil eraser, or about the size of a small stud earring. In the imperial system, that's less than 0.2 of an inch. It would fit comfortably on the tip of your pinky finger with room to spare. This is not a creature that commands attention through size or intimidation. it succeeds through something far more subtle. The jellyfish has a bell-shaped body, smooth and dome-like, with a quality of transparency that makes it seem almost like animated glass drifting through the water. If you were to look closely at one under proper lighting, you'd see its bright red stomach visible through that transparent body, a splash of color in an otherwise crystalline form. The red is due to the contents of its digestive system, catching light and announcing that yes, despite appearances, this is a living, breathing, eating organism. Young Turritopsis start their free-swimming life with just 8 delicate tentacles trailing from the edges of their bell. But as they mature, they can develop many more. A fully mature adult might have anywhere from 24 to 90 of these tentacles, though the number varies dramatically depending on where in the world they live. Researchers have discovered that Turritopsis in tropical waters like those around Panama tend to have only about 8 tentacles even as adults. But those living in more temperate regions such as the Mediterranean Sea and the waters around Japan often develop the full complement of many dozen tentacles. Why this difference exists is still not entirely clear. Temperature might play a role. Food availability might matter. Or it could be a response to different environmental pressures in different ocean ecosystems. These tentacles aren't just for show. They're weapons and tools, equipped with specialized stinging cells called conidocytes. Like all jellyfish, Turritopsis is a cnidarian, a member of an ancient phylum that also includes sea anemones and corals. Those stinging cells contain tiny harpoon-like structures that can inject venom into prey. For a creature this small, the prey consists mainly of microscopic organisms, tiny planktonic creatures, fish eggs floating in the water, small mollusks, and other bits of nutritious material drifting through the ocean. Turritopsis is a carnivore, a predator despite its delicate appearance. It drifts through the water, tentacles spread like a net, and anything small enough that brushes against those stinging tendrils becomes dinner. Originally, scientists believed Turritopsis dohrnii was native to the Mediterranean Sea, where it was first formally described back in 1883. But in the decades since, and especially after its unusual life cycle became known, researchers started looking for it more carefully in other parts of the world. What they discovered was startling. Turritopsis dohrnii is now found almost everywhere. It lives in temperate and tropical waters across the globe. It's been documented in the Indian River Lagoon in Florida. It's found on both the Caribbean and Pacific coasts of Panama. It thrives in Japanese waters. It's appeared in harbors, marinas, and docks worldwide. How did such a tiny creature spread so far? The answer appears to be us. Ships traveling the world's oceans carry jellyfish polyps on their hulls, tiny hitchhikers clinging to the metal and fiberglass. When those ships travel from port to port, the polyps travel with them. And because these polyps can produce medusae that can then revert back to polyps if conditions aren't ideal, they have remarkable staying power. They can survive journeys that would kill other organisms. This has led to what scientists call a silent invasion, a species spreading across the world's oceans not because it's harmful or noticeable, but simply because it's extraordinarily resilient. Unlike some invasive species that cause ecological havoc or economic damage, Turritopsis seems to exist quietly wherever it spreads. It doesn't disrupt ecosystems dramatically. It doesn't outcompete native species in obvious ways. It simply shows up, settles in, and becomes part of the local plankton community. This low profile may be one reason its global spread went unnoticed for so long. It's not causing problems, so no one was tracking it carefully until scientists became interested in its immortality. To understand what makes Turritopsis so special, we first need to understand what's normal. So let's walk through the typical life cycle of a jellyfish. The path that nearly every other species follows, the path that ends in death. It begins with reproduction. Mature jellyfish, whether they're the size of a thumbnail like Turritopsis, or as large as a washing machine like some deep-sea species, produce eggs and sperm. In most species, these gametes are released into the water where fertilization occurs externally, though some species have more complex mating behaviors. Once an egg is fertilized, it develops into a tiny, free-swimming larva called a planula. Picture something smaller than a grain of rice, oval-shaped, covered in microscopic hairs called cilia that beat in coordinated waves to propel it through the water. The planula drifts with currents for a period of time, anywhere from hours to days depending on the species and conditions. It's searching, in a sense, though. It has no brain and no conscious thought. It's responding to chemical cues and textures, looking for the right place to settle down. Eventually, when it finds a suitable hard surface, a rock, a dock piling, a ship hull, a shell, the planula attaches itself. This is a momentous decision in the tiny creature's life, though it makes the decision through nothing resembling conscious choice. Once attached, it metamorphoses. The attached planula transforms into a polyp, which looks nothing like the jellyfish form we typically picture. A polyp is a tube-shaped organism, usually just a few millimeters tall, attached at one end to the surface and with a mouth surrounded by tentacles at the other end. It looks more like a tiny sea anemone than a jellyfish. The polyp feeds on passing plankton, growing and developing. In many species, a single polyp will produce more polyps through asexual budding, creating a colony of genetically identical individuals all attached to the same surface. Then comes the transformation that most of us associate with jellyfish. The polyps begin to produce medusae through a process called strobilation. The polyp essentially creates miniature jellyfish from its own tissue, which bud off and swim away as free-living organisms. These tiny medusae drift with ocean currents, feeding and growing until they reach sexual maturity. At that point, the cycle begins again. They produce eggs and sperm. Fertilization occurs, planulae develop, and a new generation of polyps forms somewhere in the ocean. For the vast majority of jellyfish species, once the medusa stage produces those eggs and sperm, its job is done. It has reproduced. It has passed its genes to the next generation. After spawning, the medusa typically dies. This is the natural order: birth, growth, reproduction, death. The great cycle that governs nearly all life on Earth. The medusa stage might live for a few weeks, a few months, or in some species up to a few years. But eventually, inevitably, it dies. The polyps, meanwhile, can live much longer. Some jellyfish polyps can persist for many years, continuously budding off new medusae. But the free-swimming adult form, the medusa, is temporary. It's the reproductive stage, and once that reproduction occurs, there's no biological reason for it to continue existing. Energy conservation at the species level favors dying after reproduction, allowing nutrients to return to the ecosystem and preventing older, potentially damaged individuals from competing with younger, fitter ones for resources. This is the pattern. This is the rule. From single-celled organisms to plants, to insects, to fish, to mammals, to humans, the arrow points one direction. You can't unmake an adult and turn it back into a juvenile. You can't reverse development. Cells differentiate as organisms grow, meaning they specialize. A heart cell becomes a heart cell. A nerve cell becomes a nerve cell. They can't just decide to become something else, some earlier form, to de-differentiate and start over. Except that Turritopsis dohrnii completely ignores this rule, and the way it does so is perhaps even more fascinating than the fact that it does. When a Turritopsis medusa encounters unfavorable conditions, something remarkable happens. The triggers can be various. Physical injury, being damaged by a predator that doesn't quite finish the kill. Environmental stress, a sudden change in water temperature or salinity. Starvation, not enough food in the surrounding water. Or simply old age, the accumulation of cellular damage over time. Any of these conditions can initiate the process. The medusa stops swimming normally. It sinks toward the seafloor or the bottom of whatever container it's in. And then it begins to transform. Its bell-shaped body contracts. The tentacles retract. The whole organism becomes compact, pulling itself inward until it forms what researchers call a cyst stage. This is a blob of tissue, a clump of cells that no longer resembles a jellyfish. To the untrained eye, it might look like the creature is dying, dissolving, breaking down. But something very different is happening. Inside that blob, at the cellular level, a process is unfolding that defies everything we thought we understood about cell biology. The cells that made up the jellyfish, cells that had specialized into nerve cells, muscle cells, digestive cells, all highly differentiated and committed to their particular roles, are changing. They're losing their specialized identity. They're becoming flexible again, plastic, capable of becoming something else entirely. This process is called transdifferentiation, and it's extraordinarily rare in nature. Transdifferentiation means that a fully mature, specialized cell directly converts into a different type of specialized cell without going through a stem cell intermediate stage. In Turritopsis, muscle cells can become nerve cells. Nerve cells can become cells that will form polyp tentacles. The entire cellular organization of the adult medusa is dismantled and rebuilt into something earlier in the developmental sequence. This transformation takes between 4 and 72 hours, depending on conditions. During this time, the cyst is vulnerable. It can't swim away from predators. It can't actively hunt for food. It's essentially a sitting target, reorganizing its entire being in a desperate bid for survival. But if it succeeds, if it makes it through those crucial 2 or 3 days without being eaten or destroyed, something emerges from the cyst that should be impossible. a polyp, not a planula larva, not eggs, not sperm, a polyp. The jellyfish has skipped backward over several developmental stages. It has rejected the finality of its adult form and returned to an earlier stage in its life cycle. This new polyp is genetically identical to the medusa it came from. It's not an offspring. It's the same individual, just younger, reset, given another chance. And from this polyp, through asexual budding, new medusae will eventually be produced. These new medusae are also genetically identical to the original. The organism has effectively rebooted itself. It can feed again as a polyp, grow again, and eventually release new medusae to live as adult jellyfish once more. And here's the truly mind-bending part. There appears to be no limit to how many times this can happen. A single Turritopsis individual can, in theory, go through this cycle indefinitely. Medusa to cyst to polyp to medusa to cyst to polyp, over and over, potentially forever. This is why it's called the immortal jellyfish, not because it lives forever by simply not dying, but because it has discovered a way to reset its biological clock whenever death approaches. It's like having unlimited lives in a video game. Every time you're about to lose, you hit reset and start the level over. The same character, the same genetic code, just rewound to the beginning. To truly appreciate how radical this is, we need to dive deeper into what's happening at the cellular and molecular level. We need to talk about transdifferentiation not just as a concept, but as a biological mechanism. In normal development, cells become increasingly specialized as an organism grows. A fertilized egg, which has the potential to become any cell type in the body, begins to divide. Those early cells are pluripotent, meaning they can still become many different things. But as development continues, cells start committing to particular fates. They turn on certain genes and turn off others. They produce specific proteins that define their identity. A cell that's going to become part of the heart activates genes for producing heart muscle proteins and permanently deactivates genes for nerve cell proteins. These decisions are supposed to be irreversible. In mammals, including humans, we've known for decades that you can take specialized adult cells and, with the right cocktail of chemicals and genetic factors, reprogram them back into a pluripotent state. These are called induced pluripotent stem cells, and the discovery won a Nobel Prize because of its implications for regenerative medicine. But that process requires intense intervention. You have to flood the cells with specific transcription factors. You have to carefully control their environment. It's artificial, laboratory-based, and even then, it doesn't always work perfectly. What Turritopsis does is accomplish something similar naturally, without any external intervention, as a normal part of its life cycle when needed. And it doesn't stop at creating pluripotent cells. It goes further. Those cells don't just become flexible and wait for instructions. They actively transdifferentiate, directly converting from one specialized type to another specialized type needed for the polyp form. Recent genomic research has started to reveal some of the molecular magic behind this process. In 2022, a comprehensive study comparing the genome of Turritopsis doharni. With closely related jellyfish species that can't perform this trick, identified some key differences. The immortal jellyfish has specific variations in genes involved in DNA replication and repair. It has enhanced telomere maintenance mechanisms. Telomeres are the protective caps on the ends of chromosomes that typically shorten each time a cell divides, eventually leading to cellular aging and death. Turritopsis appears to have ways of maintaining telomere length during its life cycle reversal, effectively resetting the aging clock at the cellular level. The jellyfish also shows elevated activity in genes related to stem cell renewal and pluripotency. During the cyst stage, when transdifferentiation is occurring, researchers have found high expression of genes involved in DNA damage response and repair. This makes sense. If you're going to completely reorganize your cellular identity, you need robust mechanisms to prevent errors, to fix any genetic damage that might occur during the chaotic process of dismantling and rebuilding. Interestingly, genes associated with aging and longevity in other animals also show unusual patterns in Turritopsis. Genes like serine racemase, which in mammals is linked to aging-related cognitive function, and MSRA, which protects cells from oxidative damage, are both highly active during the cyst stage. It's as if the jellyfish is not just reorganizing its structure, but actively rejuvenating itself at the molecular level, repairing accumulated damage, resetting systems that had begun to wear down. Some of the most fascinating discoveries involve genes that were previously identified as crucial for maintaining pluripotency in other organisms. The Yamanaka factors, named after the scientist who discovered them in the context of creating induced pluripotent stem cells, are a set of genes that can reprogram adult cells back to a stem-like state. Turritopsis appears to express similar genes during its transformation. The organism has essentially evolved its own internal reprogramming system, a biological toolkit for cellular youth that it can deploy whenever needed. It's worth noting what transdifferentiation means for the broader field of medicine and biology. In humans, transdifferentiation happens, but it's usually associated with disease, particularly cancer. When cells start changing their identity inappropriately, losing their normal differentiation signals and becoming something they shouldn't be, that's often a sign of cellular dysfunction. Tumors can arise from this kind of uncontrolled cellular transformation. But Turritopsis manages controlled transdifferentiation without developing cancer. It reorganizes its entire body, billions of cells changing their identity in a coordinated fashion, and comes out the other side as a healthy, functional polyp. Understanding how it achieves this, how it activates the cellular flexibility needed for transformation while avoiding the chaos of uncontrolled growth, could have profound implications for regenerative medicine. If we could learn to trigger safe, controlled transdifferentiation in human cells, we might be able to regenerate damaged organs, repair spinal cord injuries, treat degenerative diseases, but we're still in the very early stages of understanding. Turritopsis is difficult to keep in captivity, which limits experimental research. The transformation process is rapid and complex, making it challenging to study in real time. and the genomic tools for working with this species are still being developed. We know some of the genes involved, but the complete network of molecular interactions that makes the transformation possible remains largely mysterious. In the entire world, one scientist has become particularly famous for his dedication to keeping Turritopsis dohrnii alive in laboratory settings. His name is Shin Kubota, and he works at Kyoto University in Japan. Kubota has managed something that most researchers have found impossible: maintaining a stable population of immortal jellyfish in captivity for extended periods of time. This is not an easy feat. Turritopsis is finicky. The water conditions must be precisely maintained. Temperature, salinity, pH, all of these factors need to be kept within narrow ranges. The jellyfish are tiny and delicate, easily damaged by handling or even by water currents that are too strong. They need to be fed specific food, typically brine shrimp cysts called artemia, and the feeding must be monitored carefully to ensure they're actually eating and digesting properly. Kubota checks on his colony daily. Every single day, without fail, he inspects each jellyfish, observes their condition, ensures they are healthy. He's watched them go through the transformation process repeatedly. Over a 2-year period that he documented, his colony underwent the life cycle reversal 11 times. 11 times, these tiny creatures faced stress or age, transformed into cysts, reorganized themselves into polyps, grew new medusae, and started again. 11 cycles of death avoided, 11 resets, 11 demonstrations of biological immortality in action. But Kubota isn't just a scientist. He's also, fascinatingly, something of a performer. He regularly appears on the Japanese television to talk about his immortal jellyfish, explaining their biology to popular audiences, sharing the wonder of these creatures with people who might never see them otherwise. And he's written songs about them. Yes, songs. He performs these songs at the end of his scientific conference presentations, singing about the jellyfish's remarkable abilities, about the mystery of life and death, about the lessons these tiny, transparent creatures might teach us. There's something touching about this, about a scientist so dedicated to his subject that he creates art in its honor. It speaks to the sense of wonder that drives the best scientific research. Kubota hasn't lost sight of the fact that what he's studying is extraordinary, that these jellyfish are not just experimental subjects but marvels of evolution, tiny miracles drifting in their tanks. His work has been crucial for the field. Without captive populations, it's nearly impossible to study the transformation process in detail. In the wild, finding a jellyfish at the exact moment it's about to undergo reversal is almost impossible. They're too small, too dispersed, and the trigger might come from conditions that aren't visible to a human observer. But in Kubota's lab, he can induce the transformation deliberately, control the conditions, observe every stage, take samples for genetic analysis. His colony has provided specimens for researchers around the world who are trying to understand the molecular mechanisms of the transformation. And yet, despite Kubota's success, Turritopsis remains challenging to work with. Very few other laboratories have managed to maintain long-term colonies. The jellyfish require constant attention, specialized knowledge, patience that not every research program can sustain. This is one of the frustrations of working with non-model organisms, species that haven't been developed as standard laboratory animals. We have extensive tools and protocols for mice, fruit flies, certain worms, zebrafish. But for something like Turritopsis, we're still figuring out the basics of care and husbandry. This limitation has slowed research progress. There's so much we still don't know about these jellyfish, so many experiments that could be done if only we had easier access to healthy specimens. But science progresses incrementally, and the work continues. Kubota and the handful of other researchers working with living Turritopsis are building the foundation of knowledge that future scientists will need. The spread of Turritopsis dohrnii around the world tells a story about the modern ocean, about how human activity reshapes ecosystems in subtle ways, and about the resilience of life when given the tools to survive almost anything. As we've mentioned, genetic analysis has revealed that Turritopsis populations across the globe are remarkably similar. Jellyfish from Panama, Florida, Japan, and the Mediterranean show high genetic similarity, much more than you'd expect from populations separated by thousands of miles of ocean. This pattern doesn't match natural dispersal. Jellyfish medusae, while they can drift with currents, don't typically travel across entire ocean basins. And polyps are sessile, attached to surfaces, completely unable to move on their own. The explanation that makes the most sense is anthropogenic dispersal. We did this. Human ships traveling from port to port, carrying polyps attached to their hulls, have spread Turritopsis around the world. It's an accidental introduction, unintentional, probably unnoticed. But the consequences are that a jellyfish that was once limited to the Mediterranean is now truly cosmopolitan, present in tropical and temperate waters on every continent. Interestingly, Turritopsis shows some adaptation to local conditions. The difference in tentacle numbers between tropical and temperate populations suggests that the jellyfish can respond to environmental variation. Whether this is genetic adaptation happening over many generations, or some form of developmental plasticity, where the same genetic code produces different forms in different environments, isn't entirely clear yet. But it demonstrates that the immortal jellyfish isn't just surviving in its new homes. It's adapting, settling in, becoming part of the local ecosystem. Should we be concerned about this invasion? In many cases, invasive species cause serious problems. They outcompete native species, disrupt food webs, alter habitats, cause economic damage. Think of zebra mussels clogging water intake pipes in North America, or pythons devastating mammal populations in the Florida Everglades. Invasive species can be ecological disasters, but Turritopsis appears to be what scientists call a silent invader. It spreads without causing obvious harm. It's too small to be a major predator of anything humans care about. It doesn't form massive blooms that clog fishing nets or sting swimmers. It doesn't produce toxins that accumulate in seafood. It simply exists, one more tiny drifting predator among thousands of species of plankton. In some harbors, it's become one of the more common hydromedusae. But this hasn't translated into measurable ecological disruption. This doesn't mean there's no impact. In ecology, subtle effects can cascade in unexpected ways. But so far, decades after its global spread was first documented, there haven't been reports of Turritopsis causing problems. It may be that its size and lifestyle simply don't position it to become a major disruptor. Or perhaps we simply haven't looked closely enough yet at the communities it's joining. Time will tell. What the global spread does demonstrate is the remarkable survival ability granted by the life cycle reversal. A polyp on a ship's hull, traveling across an ocean, might experience all kinds of stress. Temperature changes as the ship moves through different latitudes. Exposure to different water chemistry. Periods out of water if the ship is in dry dock. For most marine organisms, such a journey would be fatal. But if Turritopsis polyps produce medusae that then face unfavorable conditions, those medusae can simply transform back into polyps and wait it out. They can reset, survive, persist until conditions improve. This resilience makes them extraordinarily successful hitchhikers. In a way, Turritopsis has found its evolutionary niche in the Anthropocene, the current geological age defined by human impact on Earth's ecosystems. We've created a world of ship hulls and harbors, of global transport networks connecting every ocean. And this tiny jellyfish, with its unique ability to reset and start over, has exploited that human infrastructure to spread across the planet. It's a reminder that evolution doesn't happen in isolation from human activity. We shape the environment, and organisms adapt to the new niches we inadvertently create. So we've established that Turritopsis dorni can, in theory, live forever by repeatedly resetting its biology. But now we need to address a crucial question. Does it actually achieve immortality? And if not, what does that tell us about the nature of biological immortality itself? The term biologically immortal has a specific meaning in science that is different from the colloquial understanding of immortality. When we say an organism is biologically immortal, we mean that it doesn't have a built-in expiration date, that aging alone won't kill it. It doesn't mean the organism is invulnerable or that it will actually live forever. This is an important distinction that sometimes gets lost in popular accounts. Turritopsis dorni is biologically immortal in the sense that it has a mechanism to avoid death from aging. When its body begins to fail, it can reset. It can restart the clock. There's no maximum lifespan programmed into its biology. In a perfectly safe environment, with unlimited food and no threats, a Turritopsis could theoretically live indefinitely, cycling through its life stages over and over without end. But the ocean is not a perfectly safe environment. In fact, it's extraordinarily dangerous, especially for something as small and defenseless as a jellyfish smaller than a pinky fingernail. Turritopsis faces constant threats. Predators are everywhere. Fish eat jellyfish. Sea turtles eat jellyfish. Some sea slugs specialize in eating hydrozoans. Larger jellyfish sometimes prey on smaller ones. For Turritopsis, the ocean is full of mouths that would happily consume it. And predation doesn't care about biological immortality. A fish doesn't check whether its meal has the ability to reset its life cycle. It just eats. If a Turritopsis medusa gets consumed by a predator, that's it. Game over. No transformation can save it from being digested. Similarly, polyps attached to surfaces are vulnerable. Crabs, sea slugs, other benthic feeders can eat them. A strong current can tear them loose. Human activities like boat maintenance can scrape them off. Disease is another threat. Turritopsis can get sick. Pathogens can infect it. And while the transformation process might potentially allow a diseased medusa to reset and start over with a healthier polyp body, this depends on the disease not killing the organism before transformation completes, or on the disease not affecting the polypy stage as well. Many parasites and pathogens in the ocean can infect multiple life stages. Environmental catastrophes can strike. A sudden plume of pollution. A dramatic temperature shift beyond what the species can tolerate. Physical disturbance destroying the substrate the polyps are attached to. These are all ways that Turritopsis individuals die despite their theoretical immortality. So, while the mechanism exists, while the potential for eternal life is there, the reality is that most Turritopsis doorni probably live relatively short lives by human standards. They drift through the plankton for some period of time. Maybe they get lucky and avoid predators long enough to reproduce or transform once or twice. But eventually, something gets them. They get eaten, they get sick, they encounter a condition they can't escape from. This gap between potential and reality is important. It tells us that immortality, even biological immortality, is not the same as invulnerability. Every organism exists in an environment full of dangers. Evolution can give you tools to survive, but it can't eliminate all risk. What Turritopsis has evolved is a way to avoid one specific kind of death, the death that comes from simply wearing out, from aging, from accumulated cellular damage. But it can't evolve away from being part of a food web, from being small and soft and tasty to predators. There's also a philosophical question embedded in all of this. If a Turritopsis medusa transforms into a cyst and then into a polyp, is it still the same individual? Genetically, yes. It's the same DNA, the same genetic entity. But what about identity? The medusa had a particular structure, particular cells arranged in a particular way. All of that gets dismantled. The cells change identity. New structures form. The polyp that emerges is genetically identical, but is it the same individual in any meaningful sense? For a human, if we could somehow disassemble our entire body, reorganize all our cells into a different form, and then rebuild into an infant version of ourselves, would we consider that the same person? Would memories persist? Would consciousness be continuous? For Turritopsis, these questions are less relevant because jellyfish don't have complex brains or continuous consciousness in the way we do. But the philosophical puzzle remains. Some scientists and philosophers argue that what Turritopsis achieves isn't really individual immortality at all. Instead, it's a form of rejuvenation that creates a renewed individual that happens to be genetically identical to the previous one. It's like the organism is its own offspring in a strange, asexual form of reproduction that skips the usual genetic mixing. From this perspective, the medusa doesn't escape death. It dies, and something genetically identical but structurally new emerges. Others argue that genetic continuity is what matters, that we should consider the entity that persists through transformations to be the same individual regardless of structural changes. After all, our own human cells are constantly being replaced. The atoms in your body today are not the same atoms that were there 7 years ago. Most of your cells have been replaced multiple times over your lifetime. Yet we still consider you to be you. If human identity can persist through cellular turnover, why shouldn't jellyfish identity persist through a more dramatic but still coherent transformation? There's no definitive answer to this philosophical question, but wrestling with it helps us appreciate what's truly unusual about Turritopsis. Whether or not it achieves true individual immortality, it has definitely broken the usual rules of biological life cycles. It has found a loophole, a way to move backward through developmental time, a reset button that no other complex animal possesses. The discovery of biological immortality in Turritopsis has captured the imagination of the medical researchers around the world. If a jellyfish can reset its cellular age, if it can reprogram adult cells back into earlier developmental states, if it can avoid the ravages of aging, might we learn something applicable to human health? Could the secrets of the immortal jellyfish lead to treatments for age-related diseases? Could they help us extend human health span or even lifespan? These are the questions driving much of the current research into Turritopsis biology, and the answers are both promising and complicated. First, let's acknowledge what's clearly not going to happen. Humans are not going to develop the ability to transform into polyp-like forms and reset our lives. Our biology is too different. Our complexity is orders of magnitude greater. The developmental program that builds a human from a fertilized egg is vastly more intricate than the one that builds a jellyfish. We can't just borrow Turritopsis genes and expect them to work in human cells. Biology doesn't translate that directly between species separated by hundreds of millions of years of evolution. But that doesn't mean there's nothing to learn. The mechanisms underlying transdifferentiation, the way Turritopsis activates cellular plasticity, the genes involved in DNA repair and telomere maintenance, these could provide insights applicable to human medicine even if the complete process can't be replicated. One major area of interest is regenerative medicine. Humans have very limited regenerative capacity. We can heal wounds, repair broken bones to some extent, regenerate liver tissue if part of it is removed. But we can't regrow lost limbs, repair severe spinal cord damage, or regenerate entire organs. Understanding how Turritopsis cells change their identity might help researchers develop better ways to program human cells for regeneration. We already have some tools in this direction. Induced pluripotent stem cells, which I mentioned earlier, can be created from adult human cells and then potentially used to grow replacement tissues. But the process is inefficient, can lead to tumors, and doesn't always produce cells with the exact characteristics needed. If we could understand how Turritopsis achieves controlled transdifferentiation, how it changes cell identity without triggering cancer, we might develop safer and more effective regenerative therapies. Age-related diseases are another target. Conditions like Alzheimer's disease, Parkinson's disease, osteoarthritis, macular degeneration. All of these involve cellular damage and loss of function over time. What if we could trigger rejuvenation in specific tissues? What if we could reset the cellular age of neurons affected by neurodegeneration, or cartilage cells damaged by arthritis? Turritopsis demonstrates that cellular age is not fixed, that the clock can be rewound. Finding ways to do this in human cells, even partially, could revolutionize treatment of aging-related conditions. Cancer research is particularly interested in turritopsis. As I mentioned before, in humans, transdifferentiation is often associated with cancer. Cells that lose their differentiated state and start changing identity inappropriately can become tumors. Yet, turritopsis goes through massive transdifferentiation without developing cancer. It must have mechanisms to prevent the process from going haywire. Ways to maintain genetic integrity during cellular reprogramming. Understanding these mechanisms could lead to new cancer prevention or treatment strategies. Researchers studying the immortal jellyfish have found that it has enhanced DNA repair mechanisms and robust systems for maintaining telomere length. Both of these are relevant to human aging. In humans, as cells divide over our lifetime, telomeres shorten. Eventually, they become so short that cells can no longer divide, entering a state called senescence, or simply dying. Telomere shortening is one of the hallmarks of aging. If we could learn from turritopsis how to maintain telomeres more effectively, it might slow aspects of aging. However, there's a catch. In humans, telomere maintenance is tricky because most of our cells deliberately don't maintain telomeres. This is actually a protective mechanism against cancer. Cancer cells need to divide indefinitely, and they do this in part by reactivating telomerase, the enzyme that maintains telomeres. If we artificially maintain telomeres in human cells, we might extend their lifespan, but we also might increase cancer risk. Turritopsis somehow avoids this trade-off, and figuring out how could be crucial. The 2022 genomic study comparing turritopsis with non-immortal jellyfish species identified several genetic pathways that differ between them. Genes involved in DNA replication, repair, and stem cell renewal showed variations. These comparative genomics approaches are powerful because they let researchers identify the specific genetic changes that might be responsible for immortality. Once identified, these genes and pathways can be studied in other model organisms, tested to see if manipulating them affects aging or regeneration. But we're still in the early stages. The genome of turritopsis is complex. We have a draft sequence, but it's not yet complete or fully annotated. We don't know what all the genes do. The regulatory networks that control when genes turn on and off during transformation are still being mapped. And the big question, which specific combination of genetic factors is both necessary and sufficient to induce safe transdifferentiation, remains unanswered. There are also practical limitations. Even if we identify the key genes and pathways, developing therapies based on that knowledge takes decades of work. We need to test in cell cultures, then in animal models, work through the safety concerns, figure out delivery mechanisms, navigate regulatory approval, conduct clinical trials. the path from basic biological discovery to medical treatment is long and uncertain. Yet the research continues because the potential payoff is enormous. Even modest improvements in human regenerative capacity, even slowing aging by a few years, even reducing the incidence of age-related diseases, these would have tremendous impact on human health and quality of life. Teratopsis dohrnii might be tiny and obscure, but it represents a proof of concept. It demonstrates that the aging process is not inevitable, that cellular identity is more flexible than we once thought, that nature has already solved problems we're still struggling to understand. In the grand scheme of biological research, studying the immortal jellyfish reminds us of the importance of exploring biodiversity. If scientists had only focused on model organisms, on mice and fruit flies and the handful of species that are easy to keep in laboratories, we never would have discovered this remarkable phenomenon. Teratopsis was found because people were curious, because they looked at obscure little jellyfish and asked questions, because they followed up on a strange observation even when it seemed impossible. How many other biological marvels are out there waiting to be discovered? How many other species have evolved solutions to problems we face? The ocean is vast. Life is diverse, and we've barely scratched the surface of understanding it. Every odd creature, every unusual life cycle, every apparent violation of biological rules is an opportunity to learn something new. Teratopsis teaches us not just about immortality, but about the value of curiosity, the importance of paying attention to the small and strange, the insights waiting in the least expected places. And now, as we drift toward the end of our journey tonight, let's step back and consider what all of this means. What does it tell us about life, about death, about the nature of existence itself? For billions of years, life on Earth has followed certain patterns: birth, growth, reproduction, death. These cycles have driven evolution, shaped ecosystems, determined which genes pass to future generations. Death has been not just inevitable, but necessary. It clears the way for new generations, allows evolution to test new genetic combinations, prevents older individuals from monopolizing resources. Death, in a sense, is part of the deal. It's the price organisms pay for the privilege of having existed. And then along comes Teratopsis dohrnii, this tiny, transparent wanderer, and it finds a loophole. It discovers that the cycle doesn't have to be one way. It evolves the ability to hit rewind, to step back to an earlier chapter and start again. It's not breaking the laws of physics or chemistry. It's not violating thermodynamics. It's simply using the same cellular machinery that all life uses, but in a different pattern, a different sequence, a way that no one expected was possible. This should give us pause. It should make us question our assumptions about what's fixed and what's flexible in biology. For decades, scientists believed that cellular differentiation was essentially permanent, that once a cell committed to a particular identity, that decision couldn't be reversed. Teratopsis proves that's not universally true. It demonstrates that with the right conditions, the right genetic programs, cells can be far more plastic than we imagined. This has implications beyond just aging research. It challenges our understanding of development, of how organisms are built from single cells into complex forms. It raises questions about evolutionary constraints. Why don't more species evolve this ability? What trade-offs does it involve? Is there something about Turritopsis's simple body plan, its relatively small number of cell types, that makes this possible? Would it work in more complex animals? We don't have all the answers yet, but the questions themselves are valuable. They push science forward. They force us to reconsider what we think we know. There's also something humbling about this discovery. Humans have long considered ourselves the pinnacle of evolution, the most advanced, the most capable. We've developed technology, built civilizations, explored space. Yet this creature, with no brain, no complex thought, no culture or language, has solved a problem that still eludes us. It has achieved a form of immortality that all our science and medicine can't provide. This doesn't mean jellyfish are superior to humans. Intelligence and complexity have their own values, their own evolutionary advantages. But it does remind us that evolution explores many solutions to the challenges of existence, and sometimes the most unexpected organisms find the most elegant answers. Turritopsis hasn't outsmarted death through cleverness or technology. It has outlasted death through a biological innovation we're only beginning to understand. And, in the end, perhaps that's the most profound lesson. Life is resilient. Life is creative. Life finds ways. The boundaries we perceive, the limits we assume, are often just failures of imagination. The impossible might simply be the not yet discovered. And somewhere in the ocean, right now, as you listen to these words, thousands or millions of immortal jellyfish are drifting through the water, carrying within their tiny transparent bodies a secret that could reshape our understanding of what life can be. They're not thinking about immortality. They're not pondering the philosophical implications of their existence. They're just living, feeding, responding to their environment, occasionally transforming when conditions require it. They're doing what evolution shaped them to do, what their genes programmed them to do, what billions of years of trial and error made possible. And that's enough. That's remarkable. That's worth marveling at, worth studying, worth protecting. Because in understanding them, we understand life itself a little better. In appreciating their strangeness, we remember that the natural world is full of wonders. In protecting their habitats, keeping the oceans healthy, preserving biodiversity, we ensure that future generations will have the chance to make discoveries we can't yet imagine. So tonight, as you drift off to sleep, perhaps you'll dream of oceans, of tiny, transparent bells pulsing through the water, of cellular transformations happening in silence and darkness, of life that refuses to end, that finds a way to continue, that demonstrates that the boundaries we perceive are not always as fixed as they seem. The atoms in your body were forged in stars. The cells in your body carry the legacy of billions of years of evolution. And somewhere in the vast genetic library of life on Earth, a tiny jellyfish has written a chapter on immortality that we're only just beginning to read. Before you go, if these explorations into the quiet mysteries of science help you find peace at the end of your day, consider liking this video and subscribing to Sleepy Science Society. It helps others find their way to these stories, one curious mind at a time. And if there are other topics you'd like to explore in future episodes, leave a comment below. There are so many wonders waiting to be shared. Sleep well. Dream of transparent bells and turning time. And remember that in a universe full of impossible things, sometimes the smallest creatures hold the greatest secrets. Good night.